picking up on the dreams motif throughout this entire talk. This is a clip from Field of Dreams. James Earl Jones here is trying to encourage Kevin Costner's character to keep a baseball field that was built in an Iowa cornfield, and told him, if you build it, people will come, Ray. They will most definitely come. I've had that philosophy here at Florida Tech for many years, and I'm continuing it. So what am I going to build? I'm going to build an array of bioreactors with enough sensors so that we can quantify both the mass and the energy balances necessary so that we can turn tissue engineering into a true engineering discipline. So what are tissue engineering's major problems? First of all, researchers don't have funding or equipment necessary to automate the tissue engineering development process. Students spend most of their time feeding cells and emptying their waste. This leads to a lot of student frustration. In fact, I've talked to several students in Dr. Kishore's group, and even my own daughter who worked with Professor Yang this past summer. This is a massive source of student frustration. In fact, we lost one of Dr. Kishore's excellent students, Madhura Najur, who you saw in an earlier slide, to the New York Stem Cell Institute because they had automated at least the feeding and waste removal, but not the in vivo imaging. Thirdly, when students make their tissue engineered samples by hand, they can't possibly make enough of them, and consequently there's a big deal when it comes to reproducibility questions. And finally, and most importantly, most researchers stop at cell growth and to some extent differentiation, and they don't deal with the fact that the cells must integrate together, i.e. form tight junctions, in order to form a tissue. So if you had an unlimited budget, how would you go about doing this? Well, you'd want to have as many bioreactors running in parallel as possible, with an automated nutrient supply, automated waste removal, periodic patient monitoring similar to that you'd see in a hospital, and you'd definitely want to have in vivo imaging at the same time. The automation is going to minimize the reproducibility issues we just talked about, and having an array of bioreactors is going to allow us to have systematic evaluation of the synthesis variables, the cell culturing variables, whether we're going to have continuous or pulsating flow rates or voltage stimulations, etc. for tissue engineering. If we used arrays of syringe pumps, that would allow for much greater control over the mechanical properties of candidate scaffolds as well. Then here's a big issue. 3D printed scaffolding fibers right now are about 30 microns in diameter. The problem is that fibers inside the human body, think of muscles for example, are typically 1 to 3 microns in diameter. Consequently, the interaction, the cellular attachment to those fibers is going to be considerably different, because cells are typically somewhere between 5 and 30 microns. The fibers inside the body normally are going to look highly curved, whereas 3D printed fibers are going to look fairly flat. And finally, one thing that almost nobody else does is a full mass and energy balance accounting. That's common for chemical engineering projects, but you generally don't see that for tissue engineering projects. Then I started telling Julia and Athela about some of the more recent changes that we're making to our curriculum. Here you see a $3,000 kit that Labsmith is allowing us to have for quote-unquote free in exchange for beta testing it and developing a reactor prototype to go along with it. This kit has a couple of syringe pumps, valve control, a couple of three-way valves, a waste tank, a couple of feed tanks, and a laminar flow visualization chip, and it can all be built and tested in 3 hours in one lab, and is going to be incorporated into the freshman nanotech lab course in April of 2018. It's been added to the Senior Chemical Engineering Unit Ops Lab 2 course this semester, and is going to be made a permanent addition to the senior first semester lab course in chemical engineering as well. Being added on a second board is a reactor that can be used for chemical, biological, and electrochemical reactions with in situ concentration analysis. LabSmith specializes in microfluidic flows that are key to lab-on-a-chip and organ-on-a-chip devices. And what we're going to build in my new automated tissue engineering test bed won't be as small as I would really like it to be, and definitely not as small as LabSmith would like it to be, but it certainly will be a baby step toward that eventual goal. When Julia heard this, she said, "I wish we had this back when I was a student." So what are the aspects of the LabSmith/Vanderbilt design that are worth emulating, and which ones should be improved upon? Well, certainly the concept of using multi-port valves, as seen in this slide, is a great idea that can be improved upon. Valco and Vici have made multi-port valves as feeds to chromatographs from reactors, a very common concept for chemists, chemical engineers, and some bio people for decades. LabSmith does one thing better than Valco Vici. LabSmith's multi-port valves can be used with much smaller diameter tubing, as small as 640 microns, for example. However, as I wanted to fund this project myself for the tissue engineering testbed research project, I found Valco Vici's 10-way valves on the next slide. I found 18 of these Valco Vici multi-port 10-way valves at $25 each, down from an original cost of approximately $1,000 each on eBay, and have gotten those working. And then I looked at Vanderbilt's 8,000. Another major feature of Vanderbilt's tissue engineering testbed design is its use of 8 syringe pumps for sending an array of feedstocks like ultrapure water, phosphate buffered saline, which is PBS, tubenical types, or salt water to the rest of us, multiple dyes, glucose, well actually Dolbeco's modified Eagle medium, or DMEM, multiple cell types, and even drugs for toxicological testing. The employment of multiple syringe pumps is a great idea that should be expanded upon. Adding even more syringe pumps can add versatility to the number of types of cells, more customized growth media, growth and differentiation factors, etc. The problem with such expansion obviously is cost. Each of LabSmith's syringe pumps cost $800. Klern sells 8-channel syringe pumps for $1,600. Klern sells these 8-channel syringe pumps for $1,600. I wound up finding them on eBay for $200 each and snapped up 3 of them. In fact, from a bankrupt biomedical engineering company. They were still unopened in the box. Sophomore chemical engineer Thomas Quaid is currently replacing the ancient Klern electronics with Arduino and National Instruments LabVIEW controls. And with this many valves and syringe pumps for so little money, we are going to out-mass produce our colleagues from other institutions. Most notably, the competition is from Vanderbilt, which you've seen, and from a combined group of Harvard and MIT types. Then I looked at Vanderbilt's $8,000 Nikon inverted microscope. We could go that route, but at least at first our bioreactors will not be that small. So either I would need to make a much larger incubated volume and a motor-controlled stage similar to what you would see for incubated bioprinters, or I would have to come up with one of several options. I frequently use $20 1000X microscopes from China, so maybe at that cost each bioreactor could have its own microscope. However, the distance from the microscope to a glass slide coated with 3D printed tissue scaffolding might be too far, and I'm sure that I can't submerge that, or for that matter, the Nikon microscope. On the other hand, if we have to image all samples with one common confocal laser scanning microscope, how would we do that? As seen earlier in the talk, dyes are used to highlight specific biomarkers of cell proliferation and differentiation. With the aforementioned multiport valves, we could easily send any one or any combination of dyes to any bioreactor or to a standalone microscope stage area. Ideally, one would like to do truly in vivo imaging, but that will have to be a long-term research project, as the vast majority of dyes used for histological or immunofluorescent staining are irreversible, and most work by solubilizing cell membranes. Development of reversible stains would be a major benefit to tissue engineering research and development. It is possible that here too nanotechnology can provide the solution. Targeted drug delivery and even genetic reprogramming is done elsewhere by attaching drugs and genes to nanoparticles, frequently either gold or silver, allowing investigators to do some simultaneous imaging and delivery as shown earlier. On the other hand, you could take a biopsy of cells from the slide of growing cells, in which case you wouldn't have to expose the entire slide to the dyes, which are, after all, potentially hazardous chemicals. This would require the following: an Arduino-based robotic arm, which would attach a disposable syringe and syringe tip to it. Then we'd use the syringe to pipette a biopsy of cells from the bioreactor to the microscope stage. That would then get exposed to the dyes, imaged, and disposed of along with the syringe and syringe tip into a biohazard bag. That's a lot of steps. An in vivo imaging option we are starting to explore is a submersible fiber optic LED microscope camera available from Atlas Scientific.